I would like to explain some answers for homework 2. In question number 1, you were asked to come out with the structure of a polysaccharide that was treated by dimethyl sulfate and then subjected to acidic hydrolysis. So, and the names for 3 products after this treatment were given to you. So this question is referred you to slide number 20 from lecture number 16. At this lecture we discussed that chemical methods can be applied to determine structures of polysaccharides, namely the linkages in the main chain and in the branch points. And to do this, scientists treat an unknown oligo or polysaccharide with dimethyl sulfate, which leads to exhaustive methylation of all OH groups in the molecule, followed by hydrolysis. And in hydrolysis product, you see all OH group remain methylated, except those that were involved in the linkage when they were part of oligosaccharide in this case. So now let's go back to our question, and we will start with drawing the structure of the 3 products that are given to you. Let's take a look at the structures of products of polysaccharide chemical treatment. There are 3 of them, and they all are glucose derivatives which differ from each other by different methylation patterns of OH groups. For example, the first one in the list has 2 OH groups unmethylated, which means that position number 1 and position number 4 were involved in binding with other monomeric units within the structure of polysaccharide, original polysaccharide. So the ratio of the first product to the other 2 is 20 to 1 to 1. So I have my ratio here. So this original polysaccharide most likely was homopolysaccharide branched structure like this. And let me tell you why. If you take a look at the structure of the middle product, it has 2 OH group methylated and 3 OH groups unmethylated. And this is exactly the branch point found in branched homopolysaccharides. Let's take a look at this branch point. This oligosaccharide, schematically shown here, is connected by 3 neighbors. This one, and this one. So if it is connected to 3 neighbors, then after hydrolysis, after treatment with dimethyl sulfate and hydrolysis, it will have 3 OH groups, this, and that, unmethylated. And 2 OH group present here will be methylated. This oligosaccharide has only 1 OH group unmethylated, and it's most likely the end is the terminal one. So you can see, for example, this schematically shown here, monomeric unit is bound by only 1 OH group to the neighboring one. So, and this OH group will remain unmethylated, while other 4 will be methylated. Okay, so we stick to this idea, to the idea that our original oligosaccharide is branched one, and based on these ratios, I come out with this structure in which we have 19 units of the oligosaccharide that is bound by position 1 and 4, 2 positions, and this structure corresponds to the product, to the first product in the list, this one. So position 1 and 4 are involved in linkages, right? Now I have the same structure here. So this is my 20th structure. So altogether I have 19 here and plus 1. So I have 20 units like that. And I have each of these units presented in my structure as a single time. This unit is a terminal unit, is a non-reducing terminus, right? That's a structure corresponds to the last in the list. And this is a branch point right here, which is bound to in polysaccharide structure by 3 atoms, 1, 4, and 6. So 1, 4, and 6 OH groups are free in this product. Okay, so this is one answer that you can give, but this is not the only one. In fact, there are several different structures that fit to these products. And for example, you can distribute these 19 units between these two positions. So you have, for example, you can have here 10, here 10. So altogether there will be 20 of them, right? So any answer that you provide and feed to the original data given will be counted as correct. Question number 2. In this question, we have to find the change in the linking number, rising number, and twist upon conversion of a 9 base pair fragment of a supercoiled DNA from the B to the D conformation. This problem refers us back to the supercoiled DNA part of our course, in which we learned that linking number is connected to twist and rising number by these relationships, and the same is true for the change in linking number, twist, and rising number. Based on this formula and these relationships, we can find the 3 parameters, and we can start with the twist, because when we talk about a helical structure of DNA, that is a characteristic. That is what characterized by the twist. So we know that twist is calculated as a number of turns in a double-stranded DNA structure, and the right-handed BDNA has positive twist. Left-handed zDNA has a negative twist by definition. Okay, now I strongly encourage you to imagine what happens when BDNA changes its conformation to zDNA. So BDNA, right-handed helical structure, should be unwound. So we have to rotate the structure counterclockwise to unwind it and make it straight. It's not shown here, but you can imagine the straight, unwound structure, 2 strands parallel to each other. Okay, and then we have to continue rotating counterclockwise to make a left turn of zDNA. So roughly we can split this process into 2 portions: unwinding of BDNA to non-helical structure, and then continuing winding it counterclockwise to get a left-handed DNA. Okay, so and in order to calculate the twist, we will calculate how many turns we are losing when we unwind BDNA, and then how many turns we gain when we make left-handed DNA. Okay, so I have minus here, minus here. So these minuses come due to the definition that when we rotate counterclockwise, we add negative turns. So this comes by definition with the fact that we rotate counterclockwise. Now 9 is a number of base pairs, and 10.4, I use this number as a number of base pairs per turn in bDNA. So the overall expression here, this term tells me how many turns we are losing upon unwinding of right-handed bDNA to all straight conformation. And then this term comes with widening of straight parallel DNA into left-handed DNA. So 9 divided by 12, it comes from 9 base pairs and 12 base pairs per turn. So when we add these two terms, we get negative 0.62. That's the change in the twist. And then we plug in this change in the twist in the equation. Delta L equals delta T plus delta W. Okay. Now, of course, we cannot solve this equation because two parameters are unknown. Let's see what is the change in the linking number. In the lecture, when I talked about supercoiled DNA, I mentioned that linking number is changed only when the integrity of double-stranded DNA is disturbed, meaning that you have to make a break in DNA, covalent break in DNA strands, and only after that the two strands can be separated from each other. This is not what happened in this process, right? According to the description, we've never had a covalent break of one or two strands. Therefore, the change in delta L equals 0. So we plug in 0, we plug in delta T, and from this expression we can find the change in rising number. So the question is delta L 0, delta T is negative 0.62, and delta W is positive. The next question takes us back to the part of the lecture where I talked about tRNA, and it asks to draw some hydrogen bonds between base pairs G psi, G, M7G, AA, and also estimate the length of the acceptor stem. Let's start with the last question. Estimate the length of the acceptor stem. So the acceptor stem, it's actually labeled here, is this portion of the molecule. And in order to estimate its length, we just simply need to calculate how many base pairs we have, and we have to follow the memorization list in which I ask you to memorize the parameters of aRNA helical structure. So in aRNA, we have 11 base pairs per turn, and we have 3.09 nanometers in length for one helical turn. Of course, here we don't have all 11 base pairs and all helical turn. But let's count how many we have. 1, 2, 3, 4, 5 full base pairs. And now, this is also acceptor stem. We have another 4 nucleotides added to it. So what I've done here, I calculated all of them as if they were the length of a helical turn, and I explain you why. But let me count, like 5 base pairs, full base pairs, and 4 additional. So altogether, I count this as a 9 base pairs. So 9 by 11 and multiplied by 3.09 nanometers, so I get this answer. Now, why I think it's legitimate to count this equal to the length to aRNA. Take a look at this 3-dimensional structure of tRNA, and you can see here the acceptor stem right here in yellow. So you can count 5 base pairs again, and then additional, in this case there are 4, equally 4. And if you take a look at this as a pattern, you can see that it's roughly corresponds by parameters a RNA. So that's why it's legitimate to have this number, to add this 4 base pairs, to add the length of these 4 nucleotides to the base pairs just for the estimate. So remember, this number is not an exact number. This is just an estimate. Okay, let's now talk about how we draw base pairs, requested base pairs. So in order to draw them, you just need to place guanosine monophosphate next to pseudouridine monophosphate and see how they can form hydrogen bonds. You need to draw at least 2 hydrogen bonds. 1 is not stable, and 3 is very rarely occurring. So normally it's 2 hydrogen bonds, and you can see that I have my NH group, hydrogen bond donating group, next to hydrogen bond accepting group, oxygen in pseudouridine, and here is NH group, hydrogen bond donating group, is next to hexacyclic oxygen at 6th position in guanine. So I just simply connect them by dashed lines, and this is my answer. So now I do the same with guanine and M7 guanine. So this is guanine methylated at 7th position. So I show here the methyl group and the positive charge on in the structure of guanine. So guanine bond. You can write, you can draw actually several different versions of GG bonds. For example, one of them can involve N7 from this guanine. But any answer is accepted as long as you have these hydrogen bonds, hydrogen bond strong, meaning that the 3 atoms that form hydrogen bonds are aligned. Okay, and AA. Here is the answer. Question number 4 asks you to work with a peptide, and the first question is to draw the peptide and indicate bonds that are hydrolyzed by trypsin and hymetrypsin. So of course, here is the answer. Of course, so you have it. It's posted on web courses. But just going back to drawing peptides, I just suggest that you draw a wiggly line, right? And the number of atoms corresponds the number of amino acids multiplied by 3. So you have to have 3 nodes, right, for each amino acid. And then you add to these nodes, you add amino groups, each third atom. Like we start with N-terminus, and then we skip 2 nodes and add amino group. You can see amino group sign trans conformation. And then you add CO oxygens, right? So this is your peptide bond, right? Peptide bond. You see oxygen also in trans conformation, which makes this actually structure stable. And yeah, and then you populate this remaining. This is alpha carbon. You populate. You add amino acid side chains, which are different for different amino acids. That's how I've drawn this structure. And then you indicate the cleavage sites, which is a simple memorization. Trypsin cleaves after positively charged lysine and arginine amino acids. So I have lysine, and I show that the cleavage takes place here and here. And I have arginine, and the cleavage takes place here and here. So right after the amino acids, if you count from N-terminus. Remember, N-terminus is the beginning. Okay, now same with hymetrypsin, which cleaves after aromatic amino acids. So tyrosine, phenylalanine, tyrosine. So 3 cleavage sites for hymetrypsin. Now, what is important here is that you do show the cleavage of a peptide bond. So the peptide bond is between CO and NH. This bond right here is not a peptide bond, and if you show cleavage of this bond, it will be counted as a wrong answer. Now let's determine the number of peaks in electrospray ionization mass spectrum. So in order to do that, you have to redraw your peptide in fully protonated form. So all groups are protonated. So the difference of this structure with the previously shown structure is minor. So basically I have my carboxylic groups protonated. The two carboxylic groups are protonated. Here they were unprotonated because this structure is relevant to physiological conditions under which trypsin and chymotrypsin cleave the peptide bonds. But this structure is fully protonated when there is excess of protons. And now after that, I count my groups. I count the full charge on this peptide, and pretty much it's counted as a number of lysine residues, arginine residues, histidine residues which are not present in this peptide, plus 1, and plus 1 stands for N-terminal amino group. So let's count 1, 2, 3, 4, 5, 6. The number of peaks in electrospray ionization mass spectrum is going to be 6 because you have an individual peak for each number of charges. You have one peak for the charge of this peptide 6. You have another peak for the charge on this peptide 5, and so on until 1. So you don't see the peptide with charge 0, even though it exists perhaps, because it's not detected by this method. Okay. Now PI. So the last question that you need to answer: what is the isoelectric point or PI of this peptide? By definition, PI of any molecule is pH under which this molecule has charge 0. Okay. So in order to answer this question, again, you work with a structure that is fully protonated. So you draw the structure in fully protonated form, and this structure is taken pretty much from the previous slide. Okay. Now what I do to the structure, I add pKa values for each of the group. So the C-terminal amino group has pKa 4. Now the glutamic acid, this pKa taken from the table, don't have to memorize it, and I populate it with other pKa's. For example, the amino group has pKa 8, and tyrosine has pKa 10.46. This is taken from the table for amino acids, and amino group of epsilon amino group of lysine has pKa 10.54. So I have 3 of them, and the last pKa appears for arginine, 12.48. Okay. After that, what I recommend you doing is listing the pKa in descending order, like this. So these are pKa's for the 2 arginine groups. These are pKa's for the 3 lysine groups. These are tyrosine groups, 2 of them, and so on. Now, what you do next is the following. So when everything is protonated at very low pH, this pH below 4, we have a total charge on the peptide 6. We just talked about this on the previous slide when we considered number of peaks in electrospray ionization mass spectrometry. And you jump Okay, so this is 6, okay? And then once you, so at pH 4, you have 50% protonated, 50% deprotonated of C-terminus, meaning that the total charge on your molecule is 5.5, in fact, because of 50%. Now, but if you jump over this pKa, right in this point, you're going to have pH, you're going to have charge plus 5. Here, plus 4, plus 3, plus 2, plus 1, and 0. So this is your PI point. And in order to calculate the exact number, you have to take average of the flanking pKa, these two pKa, 10.54 and 10.54. The average is obviously 10.54. Yeah, this is your answer. Question number 5 deals with sequencing of peptides. It refers you back to peptide sequencing and method of overlapping fragments. A given peptide, a given protein, was digested by two different methods, and you have to determine which methods we used, and then you have to assemble the full protein out of data given. Now, if you take a look at the method A, you will see that each peptide is ended by E, 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 E. The only one is not ended by E. This is R. So a protein or peptidase that cleaves after glutamic acid is V8 protease, and that's your answer, V8 protease. You have to memorize this for this course. And the R, which is not E, is most likely the terminal, the C-terminal amino acid, okay, that wasn't cleaved. Okay, now let's take a look at the method B. Again, you see methionine, arginine, methionine, all methionine, but this arginine. So if you take a look at YR, you will see YR is here. So this is definitely a C-terminal portion of your peptide. So this is like all this peptide, long peptide, is a C-terminal peptide. Now, how do you assemble them? The strategy is the following: you take let's say the end of any given peptide, for example LPAE, and you look for this sequence in the second method. And if you take a look, you will find the only one unique place where you find this sequence, LPAE, and you see it's followed by FTPA, right? So you should look for a peptide. So when you used the V8 protease, it obviously cleaved here, right? So there should be in this method A peptide that starts with FTPA. Let's take a look. You definitely see it, right? FTPA. What it means? It means that this peptide will be followed by peptide number 3, FTPA. So you should place it here. So, and by doing this thing several times, you'll assemble the entire peptide shown here.